Hello biology lovers. Today I am going to start with a topic that is sexual reproduction in flowering plants. So without wasting any time, let's start with the topic. Hello biology lovers. Today I am going to discuss a topic which is sexual reproduction in flowering plants. So what is sexual reproduction? In flowering plants, that means the plants they produce flowers and the seeds are enclosed in a fruit. So these plants they mainly reproduce by the fusion of their gametes. So in the case, the flowering plants, they reproduce with the help of syngamy. Let's understand what is syngamy. Syngamy refers to fusion of one motile small male gamete with non-motile large female gamete. So, so the whole process can be divided into 5 stages. The stages includes pre-fertilization changes, the main process, fertilization. Next, the post-fertilization changes, seed and fruit formation, and last but not the least, the special cases or special forms of fertilization. Now, let's understand with the very first part what is pre-fertilization changes. So, as I have referred that pre-fertilization changes. So, the plant has to prepare itself for the fertilization. Now, the pre-fertilization changes include fusion of gametes. Sorry. Now, we will understand about the event pre-fertilization changes. Pre-fertilization changes refers to the changes occurring before fertilization. So, before fertilization, what will occur? The formation of male and female gametes and pollination as well. Pollination, what is pollination? The transfer of pollen grains from the anther of the androecium to the stigma of the gynoecium. Now, the first event that will occur is sporogenesis. Now, what is sporogenesis? Formation of spores. Formation of spores are of two types. One is microsporogenesis and another one is megasporogenesis. So let's understand. Here, sporogenesis is of two types, microsporogenesis and megasporogenesis. Now, microsporogenesis, it occurs in the anther of the androecium. androecium is the male reproductive part of plants. Next, gynoecium. Gynoecium is the female reproductive part of the plants. Megasporogenesis, it occurs in the ovary of gynoecium. What it really forms? It forms the megaspores, which will finally form egg. Another one, microspores, which will form pollen grain. Now, the thing is, pollen grain is not the cell which will fertilize the egg. What will happen? Pollen grain is the male gametophyte. It carries the male gametes. So, the male gametes will eventually form from the pollen grains. We will study about how pollen grains they change into or form the male gametes. The male gamete formation finally occurs in the stigma of the gynoecium after the pollination. We will study this later on. Now let's understand the further processes. Now let's understand about the male reproductive unit. Male reproductive unit is known as stamen. Stamen contains two parts. One is anther and another one is filament. The anther is the part that contains the pollen grains, or the pollen grains are formed in the anther. Anther has a very specific structure. So we will study with the structure first. After that, we will start with microsporogenesis process. So now, the anther, it is divided into two main parts. We can say anther wall, and another one is microsporangium. Anther is a typical bilobe structure which contains four microsporangiates. So, it is called a tetrasporangiate. It is called a tetrasporangiate. It is called tetrasporangiate. Now, coming to the anther wall is divided into four walls, or protected by, anther is microsporangium is protected by four walls from the outside to inside. It is epidermis, endothecium, next is middle layer, and the most important, tapetum. Now let's understand their function. Epidermis is the outermost, most protective layer. It is outermost, protective, and single cell layer. Another one, endothecium. It is inner to the epidermis, but outer to the middle layer. So endothecium, it helps in dehiscence of the anther. Dehiscence of the anther during pollination. The middle layer, it is another protective layer. It doesn't have any specific function, and it supports development and growth. Now, the most important layer, which is tapetum, it is more important than protection to nourishment. So, it is the most important tissue or cell layer for nourishment of the developing pollen grains. Now, what is microsporangium? Microsporangium is the tissue where microsporogenesis occurs. Microsporogenesis occurs in microsporangium. Now, let's understand with the label diagram of the anther in transverse section. So, this is a structure of anther in transverse section. The anther, it looks like a bilobe structure. This is the anther and this is the filament. Together they are called stamen. So this is the stamen. Stamen is the unit of androecium. So now this is whenever we cut the anther like this, it will look like this. It is bilobe, but 4 chambers of sporangium are there. So these are 4 chambers. I have drawn in detail a single sporangium. So from the outside up to the inside, we can see epidermis. It is one layer, as I have said. Inner to this, it is an endothecium, the endothecium, the blue cell layers. Inner to the endothecium, the green is middle layer. Middle layer, they help in growth and development. The another one that is red in color, here you can say, is tapetum. Tapetum, they help in the nourishment of developing the pollen grain. So these pollen grains, they will get nourishment from the tapetum layer. So these are sporogenous tissue. This black tissue having nucleus, you can see, they are sporogenous tissue. This tissue will grow into microspore mother cell, and microspore mother cell will undergo meiosis to form microspore, or which will under maturation grow into pollen grain. Now let's see the main process of microsporogenesis. Microsporogenesis. Now, this process, it occurs in the microsporangium or pollen sac. Microsporangium, or it is also known as pollen sac. So this microsporangium, it contains some pollen mother cell which are actually diploid. This pollen mother cell or PMCs, it will undergo meiosis and eventually form pollen grains. Pollen grains, they stay in tetrad form, and when they come out from the anther, they become individual and go to the stigma of the gynoecium. Now let's understand. It is 2N, as I said. It has some special tissue which is known as sporogenous tissue. This sporogenous tissue is also diploid. Sporogenous tissue, they contain pollen mother cell. In short, they are written as PMCs. PMCs are also diploid. This PMC, they undergo meiosis. Meiosis, I hope you all know what is meiosis. Meiosis is the reductional division in which the ploidy of the daughter cells becomes half to the mother cell. Now, this when the meiosis will occur, the pollen grain or microspore will be formed. In every meiosis, whenever there is a one cell involved in meiosis, it will form 4 microspore which are haploid. These 4 microspore, they form tetrad, or they are known as spore tetrad. It is then separated and forms individual microspore. Under maturity, these microspores, they form pollen grain. Now, let's understand things about pollen grains. Pollen grain. Whenever pollen grain is mature, it forms 2 parts. The pollen grain has 2 main parts, the wall or protective part or the cytoplasm. This wall has 2 parts. One is exine and another one is intine. So this is exine is outer and intine is inner. Now exine again has 2 parts. One is sporopollenin and another one is germ pore. Here intine, it is made of cellulose and covers the cytoplasm. Now let's understand what is sporopollenin. Sporopollenin is a polysaccharide, lipid and some sort of protein which is a protective layer before the protective layer around the exine. So exine, it protects the cytoplasm of the pollen grain and intine as well. Now this, what is the need of this sporopollenin? Sporopollenin provides the resistant nature of the pollen grain. Pollen grains, they can survive heat, UV radiation, sometimes some specific chemical reactions. So this nature of pollen grains is provided by this nature of pollen grains is provided by sporopollenin. Now let's understand the structure of a pollen grain. If this is a pollen grain, pollen grains has various cultures outside the exine. This is the exine which is cultured, and this inner layer, it is intine. So now you can see some pores are present here which helps the pollen grain to form pollen tube during fertilization. So this germ pore, this is known as germ pore. This germ pore helps in the pollen tube to come out during fertilization. Inner to this, it has cytoplasm and nucleus. This is cytoplasm and nucleus. Now, this pollen grain, this is the structure of a pollen grain during the maturity or before pollination. After pollination, the pollen grain will eventually grow into male gametes. We will understand the formation of male gametes after we study pollination. So let's start with we have done microsporogenesis. Now let's start with megasporogenesis. Before starting with megasporogenesis, I have to let you know about female reproductive unit, which is also known as pistil or carpel. Pistil or carpel. So let's understand what it is really is. Now, this is the stigma. This is style and this is ovary. Inside the ovary, there is it is ovule. These are various, in many ovules in ovary. It can be one or it can be many. So ovary is the main place where the female gametophyte formation or megasporogenesis occurs. So let's understand with the structure of ovary. Inside the ovary there is ovule. So ovule is the main female reproductive part which contains female gametophyte. Here I have drawn a ovule which is anatropous. Anatropous, as you can see, the structure is kind of inverted. We will have a brief video separately about the types of ovule the angiosperms have. Mainly there are uh, 4 types of ovule which can be found, but in most of the cases the ovule is anatropous in nature. Here are various structures starting from the funicle. Funicle is the stalk that joins the ovule with the base or wall of the ovary. So it has a function that is joining of ovule with ovary wall. Next is hilum. Hilum is the connecting part or the joint between the ovule and the hilum and the funicle. Next, integuments. In most of the cases, angiosperms have 2 integuments. One is outer, I have drawn in red color, and the other is green color, inner. So these integuments, they have only one function, that is protection of the embryo sac. Protection. It can be 1 or it can be 2. Next, the nucellus. Nucellus is the nourishing tissue present in ovule. This nucellus provides the required nutrients to the growing embryo sac. So, nutrients are provided by nucellus. So, it has function nourishment. Now, I have also written here embryo sac. Embryo sac is the main part that the ovule is protecting or the ovule is made for. So this is the main embryo sac, or it is also known as female gametophyte. Female gametophyte. I have written here. So embryo sac is the female gametophyte which is made of different nucleus and cells. You can count 1, 2, 3, 4, 5, 6 and the total 7 cells, and it is 8 nucleated. 1, 2, 3, 4, 5, 6, 7, 8. So embryo sac is a 7-celled 8 nucleated structure which is responsible for the main reproductive part of the females. So embryo sac, it possesses the ovum or egg, possesses egg, or the fertilization occurs in the embryo sac. Now, other than embryo sac, we have 2 structure known as micropyle. From the micropyle is the small pore from which the pollen tube comes to the embryo sac. So, it is the pore which helps the pollen tube to enter embryo sac. Opposite to the embryo sac, there is chalaza. This chalaza is the other structure or any structure that is present opposite to the micropyle. Chalaza also helps the pollen tube to enter the embryo sac. So it also helps the pollen tube to enter embryo sac. When the embryo sac enters from the chalaza, it is known as chalazogamy. But when the embryo sac is penetrated from micropyle side, it is known as porogamy. So there are main two types of entry of pollen tube. So this is known as chalazogamy and porogamy. Now let's discuss about the female gametophyte production or formation of female gametophyte or megasporogenesis. When I say megasporogenesis, it means formation of megaspores and eventually the formation of embryo sac. Now the process is ovule. It is a diploid structure. We all know, we have drawn a label diagram of ovule right back. So it is also 2N. We have megaspore mother cells. MCs. So these are 2N. When this undergoes meiosis, it forms 4 megaspores. Among these megaspores, they may form this kind of structure. They may form a linear tetrad or can be a 3D structure made. So from under this condition, the 3 of the megaspores will degenerate and only one will function as functional megaspore. So I am taking this one to be functional. So if this is the functional, the rest 3 will degenerate. It will degenerate. Now this functional megaspore, it is haploid. It will go through 3 consecutive mitotic division and form embryo sac. This embryo sac, as I said, will have 7-celled structure and 8 nucleus. Sorry, the nucleus spelling is wrong. It will have 8 nucleus. So now let's have a brief look over the embryo sac. Embryo sac is a oval structure in which we will find different types of cells and nucleus. Here I am considering this part of the ovule to be micropyle and here it is telaza. So towards the micropyle there will be 2 cells and 1 bigger cells. These 3 cells can be differentiated. Here we have 3 undifferentiated, 3 same color, same texture cells, and here in the center we will find 2 nucleus. Let's understand their names and functions. This is the egg cell. These 2 similar looking cells, these are synergids. These 3 cells, they are antipodal cells, and these two are pollen nuclei, or we can also call them secondary nucleus. Here these synergids, they helps in the fertilization process during the fertilization, and these antipodal cells, they also help in the process of fertilization. The special feature which will be revealed in the upcoming videos, we will know how the polar cell is very important in angiosperms. So this is all about the sporogenesis process of pre-fertilization. In the next video, we will understand about the pollination, their types and their agents. So stay tuned with me and follow my videos. We will understand every part of the life cycle, like from cell to the system. So beware of the classes and stay tuned.